video, we'll discuss about the asexual reproduction and its type. But before that, let us know about reproduction and its type. Reproduction is classified into two, sexual and asexual mode of reproduction. Sexual reproduction is a type of reproduction that involves two parents, that is, the fusion of male and female gametes. While asexual reproduction is just the opposite. Asexual reproduction is a type of reproduction in which only a single parent is involved. Examples of asexual reproduction are hydra, sea star, strawberry, archaebacteria, eubacteria, euglena, paramecium, yeast. While the examples of sexual reproductions are chickens, lobsters, sharks, humans, butterflies, and many other animals. Let us discuss about the types of asexual reproduction. Asexual reproduction is classified into 7 types: fission, fragmentation, regeneration, budding, vegetative propagation, spore formation, and tissue culture. First one is fission. Fission is classified into 2, binary fission and multiple fission. From the name itself we can see that it is the process in which a parent cell splits into 2 daughter cells. But in a multiple fission, a parent cell is divided into multiple daughter cells. But in binary fission, it is 2 daughter cells, and the difference is that in multiple fission, instead of 2, multiple daughters are splitted. The examples of binary fissions are amoeba, paramecium, euglena, while the examples of multiple fission are sporozoans and algae. In this picture, we can see the algae's fission, binary fission in algae, and the multiple fission in plasmodia. In algae, the parent cell divides into 2 daughter cells. The first step is the cell division, then it gets split into 2 new daughter cells. While in the plasmodium, the nucleus, which contains many daughter cells, will get split or released into multiple daughter cells. The second type is fragmentation. It is a form of sexual reproduction in which an organism split into fragments. We can see in the figure in Spirogyra, it is split into 2 parts. So this is meant by fragmentation, and each fragments, that is each pieces that are broken, will be matured into a new individual, just like their parents. Examples are filamentous, cyanobacteria, molds, lichens, sponges, aquaplaque worms, some annelid worms, and sea stars. Next one is the regeneration. It is the process of getting back a full organism from its body part is called regeneration. Example in planaria. From the figure we can see that the planaria, first it is divided into 3 parts, and from each part it grows into a fully organized individual like its parent. Next one is budding. Budding is a small part of the body organism that grows as bud, then detaches and becomes a new organism. This is how budding takes place. For example, in hydra. From the figure, in the first picture, we can see that there is a small outgrowth of bud at one end of hydra. And in the second, we can see that it grows developmentally. It grows gradually. And when it grows completely into a fully matured hydra, it detaches from its parent body. Fifth one is vegetative propagation. We are all familiar with the leaf called bryophyllum. The bryophyllum contains many leaves at its margins. When these leaves fall into the soil, they grow into a new plant. This is called vegetative propagation. It is a form of asexual reproduction occurring in plants in which new plants grow from the fragments of the parent plant, that is, from the different parts of the parent plant, that is, from leaves, roots, stems, new plants can be grown. This is called vegetative propagation. We are all familiar with the grey coating over the bread when it cooks. It is actually called the rhizopus or the bread mold, which is made up of thread-like structures called hyphae. These hyphae has sporangium, in which spores are present. These spores are the reproductive part of sporangium. These spores are covered in a thick layer. This thick layer will be removed when it comes in contact with the moist surface. So, when this thick layer comes in contact with moist surface, the thick layer is removed and the spore falls on the moist surface. And when that happens, each one forms into a new individual. Next one is the tissue culture. Tissue culture is a method of biological research in which fragments of tissues from an animal or plant are transferred to an artificial environment in which they can continue to survive and function. It means that cells from the growing part of the plant are removed, or from the growing part of the animals are removed and placed in an artificial medium where they divide and form small groups of cells. When they form small groups of cells, they are transferred to another medium containing hormones for growth, and then when they are completely grown, they will be placed into the soil for further development. This is called tissue culture. The advantages of this is that the plants grown in such a medium will be grown in a disease-free condition, and the method is used for ornamental plants too. From the figure we can see that a growing part of the plant is cut and kept in a medium where hormones are provided for the growth, and then when it grows, it is placed into the soil for further development.